My name is Vincent Bett. I'm a PhD student at Institute of Science and Technology, Austria. And today I'm going to tell you about one of the chapters of my PhD, and it's about how epigenetics may regulate sex-biased gene expression and mediate dosage compensation in Artemia franciscana. And the model organism which I'm working at is in this image. So the slides are not moving. So sexual dimorphism is quite common in animal kingdom, and this can be in form of shape, height, and even in terms of the coloration. And even in terms of behavior, physiology. And this, despite the fact that male and female share similar genome, but in species with sex determination, with genetic sex determination, the number of genes may only differ on the sex chromosome. And the sex chromosomes are acquired from the ordinary pairs of autosomes after emergence of sex-determining gene. And this is followed by the loss of recombination, and this is normally accompanied by accumulation of repeats, transposable elements, and eventually lead to degeneration of genes. And if the genes which are part of this chromosome which has lost part of it while interacting with the genes in the autosome, this can have a negative effect on the fitness on the organism. And a number of, like in ZW system, we have two types of how this imbalance can be mitigated. One, it can be in form of only the genes which are dose sensitive can be compensated. And we call it partial dosage. And around 20 to 30% of the genes in chicken, snakes are dose sensitive. But until recently, we used to believe that in CW system, only partial dosage compensation exists until this complete dosage in moth and butterfly. And this model organism which I'm working at is Artemia. It's a crustacean species and live in hypersaline environment. And it can reproduce to produce live offspring. At the same time, it can produce cysts depending on the environmental condition. And it's a ZW system. And when we started this project, we didn't have a chromosome level assembly, so we generated. And we identified the region of the genome, of the Z, which no longer combine with the W. And this region can be identified using this coverage. And you can see the drop in coverage. You have one copy in female and two copies in male. And when we look at the expression level in this region, which no longer combine between the Z and W, we see the expression to be equal in male and female, despite the gene copy number difference. This suggests that this particular species has acquired complete dosage compensation. So we were interested in this study, how do these species achieve equal expression of genes between sexes and between autosome and the sex chromosome, despite the gene copy number difference. And we have, it has been described in other organisms that it can exist in two forms. It can be in form of upregulation of transcription on the heterogametic sex, where we have one copy, so as to balance the two copies in the other homogametic sex. And this has been shown in Drosophila. In other cases, it can be in form of downregulation, where it can exist in two forms. Like in mammals, it can be in form of you entirely inactivate the entire chromosome, or it can, in nematode, like C. elegans, instead of inactivating the entire X chromosome of the two homogametic sex, you downregulate by half. And those two types which I described is always accompanied by changes in chromatin. Like when you look at the inactivated X chromosome in the female in mammals, that inactivated X chromosome normally is heterochromatic. say that like it has excess of these repressive histone marks and inactive and lack of active histone mark. But in the case of Drosophila, where we are supposed to upregulate the transcription, it's almost euchromatic, where we see excess of active histone mark and depletion of the repressive histone mark. So we were interested in looking at this epigenetic. So we generated a number of histone modification data sets that comprise of both active and repressive one, and it targets different parts of the gene body. And also we generated from heads and gonads using this particular technology called cut and tag. And cut and tag is similar to chromatin by immunoprecipitation, but the only difference is this one is based on enzymatic, where it uses this enzyme called transposase, and it cut and integrate adapters to region of the chromatin of interest. And because this is enzymatic and is non-aggressive, the signal is very high and the background noise is very low. So you require only less sequencing depth. But it may appear simply like this, but it requires a lot of optimization of the protocol. So we looked individually of those histone marks, which I listed before, to try to see which particular histone mark is enriched or in that particular region which no longer recombine in comparison to autosome between male and female. And when we look at the histone mark which target the promoter site, we didn't see statistical difference. And the one which target the enhancer site, we didn't see statistical difference. The one for the transcribed region, the same case. We also checked the repressive histone mark, we didn't see much of a difference. But when we go to H4K16, which has also been described in Drosophila to be mediating the dosage compensation, we see in males which have 2 copies, we see a similar distribution in terms of the enrichment of the genes in that particular region which no longer recombine, and also in the autosome in males. But we see upregulation or the enrichment of this particular active histone mark in the heterochromatic sex which have 1 copy. This suggests that the accumulation or the enrichment of this active histone mark is to enhance the gene expression of that particular region, so as to balance expression in the other 2 and also in the autosome. So we were interested to see whether this enrichment, does it occur only on the transcription start site or across the gene body? And when we checked it, the enrichment is not only on the transcription start site, but even across the gene body. We also checked across the genome, and we see the enrichment only on this region which no longer recombine between the Z and the W. To summarize the first part, to answer the question, what type of dosage compensation exists in atemia, is similar to what is happening in Drosophila, where we see enrichment of active histone marks so as to enhance the transcription, and this is similar to Drosophila. Okay, so after telling you about this, I also looked at whether epigenetics modification may regulate the regulation of sex bias genes. Here I'm going to go through it quickly because it's a bit complex. So we looked at differential expression between the genes in the heads and gonads, between male and female. And this particular type of species also is, as I forgot to mention, that is highly dimorphic, in which the male has these clappers that they can use to attach to females during mating. And I used the kind of data set, but instead of looking individually these histone marks, I combine these histone marks and look at the presence or absence of these histone marks across the gene body. And we use this combinatory which called Chrome HMM. But because we were interested with how the presence of these histone marks or absence at the gene level, and the gene can have multiple histone marks, we classify further this particular classification. And we use, based on functional categorization, like if a gene contains only one particular histone mark, based on this, what I described on the left part side, we consider that to be one unique chromatin signature. And we go until we classify around 16 chromatin signatures. And this particular approach is what we adopted, what was published in 2022 by Bueno et al. And so we looked at the genes which we classify to be active associated and repressive associated. And we said those which we consider to be active associated in terms of chromatin signatures had a higher expression compared to those which we classify as repressive associated. And we were interested to see like whether the genes which have differential chromatin state between the sexes, can it also have differential gene expression pattern, or are they enriched with differential gene expression? So we grouped those these particular chromatin signatures in male and female, those which contain only in male, like those which in male and female, those which contain active associated, those which have differentially, those which had contrasting chromatin state, and those which had both repressive associated. And when we looked at transcription abundance of this, which we consider to be both active in male and female, they had a higher expression compared to those which we consider to be repressive associated, suggesting that our classification made sense. And when we look at the statistical difference between those genes which had both active associated chromatin signatures, we didn't find statistical difference. But those which had contrasting chromatin signatures, they had statistical difference, suggesting that chromatin landscape may play a role in regulating this, and this may play a role in regulating the difference in transcription pattern. We look further to see how the enrichment of these chromatin signatures, contrasting chromatin signatures in genes which we consider to be male biased, unbiased, or female biased. And we say, when we compare those enrichment, we see to be statistically different. And with that, I would like to, because of time, I would like to summarize and by giving you a takeaway message that we see epigenetics may play a role in regulation of sex bias genes, and epigenetic modification do happen in female, which is a ZW system in this organism. With that, I would like to thank my amazing supervisor, Beatrice Vizoso, and my committee member, Daniel Silverman, and my group and funders. Thank you so much for listening. I think I have 1 minute for one question in case. Okay, thank you so much, and next.
Hi everybody, I'm Nathan. Thanks for coming to this room. That's very nice for me. I am at the University of Victoria in the Owens Lab. Greg's here watching. Thanks for coming, Greg. I am a master's student there, and this is a little presentation on some of my work, which itself is a little chunk of a joint project between Fisheries and Oceans Canada, NOAA Fisheries, us at the University of Victoria, and Berkeley, which is looking at Pacific rockfishes. First, I'm going to tell you a little bit about the genus that I work in, Sebastes. Those are my two guys up there, Copper and Quillback. We'll get a little bit more into them as I move along. But the genus itself, highly speciose in the Pacific. 102 species described, potential for more to be described as they are found. They can be very long-lived. There's a huge range of lifespan in these fishes, some as little as 10 years, some over 200 years old. And these two things make the genus interesting to study from an evolutionary standpoint, but also from a lifespan standpoint. Some of the work that's been done and published has been on longevity, genes associated with longevity in these fishes. I focus on something else. Among fishes, they're a little bit unique for their vivipary, internal fertilization, giving birth to live young, as opposed to the maybe more typical spray and pray approach employed by most fishes. And at least in Canada, they represent an important traditional cultural food source, contemporary food source, and are of commercial value due to their targeting by recreational fishers. They don't necessarily know what they're catching down there when they put their line 300 meters deep, but sometimes they come up with the threatened ones and sometimes not. Anyhow, these fishes, copper and quillback rockfishes, have been my focus over the past couple of years. Part of the onus for my work is to better describe the genetic diversity within quillback, as managers want to know if their current conservation units are sufficiently granular, if they capture the true genetic diversity, any private alleles and that sort of thing that may be held by these populations. And while quillback have been recognized as threatened due to overfishing in BC for about 15 years, managers in California made a similar call just last year. So I'd like to take you on a quick journey through some of my thought process, some of my master's research, looking at these two fishes, and hopefully you'll enjoy this story. What have I done? Something I'm sure each of us has asked ourselves at some point. Describe population structure within each of these species. Started with some big picture population structure analyses to identify any populations that have unique genetic variation. This got at my first question of whether the current management regimes in BC were biologically relevant. Next, I would run some pairwise tests of differentiation, looking ideally for shared variation across both species, spoiler alert. This would help me identify any hotspots in the genome, maybe point to some adaptive significance. Take a look under those peaks to see what was going on from a structural standpoint, what structurally might might drive the ability for these regions to differentiate. And then I whipped up some maximum likelihood phylogenies to understand whether or how any of the putatively important variation was shared between these species. Sampling was a bit of a mixed effort. We had some come in from both Canadian and American fisheries managers. Many were taken during annual surveys. Others were individually caught. Given that these fishes are known to hybridize, we did our best to ensure that the samples that I was using were pure types and not clear phenotypic hybrids, although to a greater or lesser extent that was true. In Canada, both of these fish are managed in aggregate with some other inshore rockfishes. And the two designated units for those are between Vancouver Island and the mainland. That's these green dots here, Salish Sea. And those outside Vancouver Island, for those of you not familiar with the coast, that's indigo, yellow, and blue there. So broad strokes population structure stuff here with PCA, this one within copper. We can see that our samples from the Salish Sea and Puget Sound cluster separately, consistent with the current management regime, so that is great. Quillback, though, have a lot less clear population structure going on. even between those fishes inside and outside of Vancouver Island, much more of a sort of panmictic cloud there. And this itself was pretty interesting to us because these species are quite closely related and presumed to have almost total overlap in their life history strategies. It is pretty hard to know what exactly is going on down there, but something is driving this difference. When I was doing my pairwise tests, because of the difference between those inside Vancouver Island and outside Vancouver Island copper, that was one of the pairwise comparisons I was interested in. But any of those pairwise FST comparisons I made looked something like this, where copper was very highly diverged across the whole genome. And Quillback had almost no differentiation across the whole thing, but for a few of these peaks, we'll call our little islands of divergence in Quillback. Unfortunately, as you can see, they're not necessarily cleanly shared between these species, but nonetheless, it made us interested in seeing what was sort of under those peaks of divergence, especially in Quillback. So we did some analysis for linkage disequilibrium. I have 5 of the chromosomes pictured here. Turns out those islands are underlain by some pretty good size haploblocks. Note that there are some commonalities here, chromosome 8, chromosome 13, chromosome 18. These blocks are in LD in both species. For chromosome 23, Quillback has a large block in linkage disequilibrium right at the very beginning. Copper does not. This type of linkage pattern was actually happening in even more chromosomes than just these, in most of them. But I pulled out a few here just to sort of demonstrate what was going on. And for the rest of the presentation, I'll use chromosome 23 as my example to dive in a little deeper and show you kind of the process I went through asking more questions. One of the programs I used to ask these questions is something called LowStruct. You may have heard of it. Basically, in little windows all along the genome, conducts PCAs within them. And I did this with both species bin together. Basically, I'm looking for differences in their combined population structure, this being the sort of normal thing. And this is actually from a chunk in the middle of chromosome 23, which represents a pretty normal pattern with copper on, copper on one side and Quillback on the other. For the most part, the major axis of variation separates the species from each other. But there are some places where the dots start to blur together. This can be for a couple of reasons. In places like centromeres, where lots of repetition and mutation and stuff sort of makes it hard for PCA to pull anything of significance out, or in highly conserved regions where the sequences are quite similar between species anyways. But there's another, possibly more interesting pattern present at the start of chromosome 23 on that LD block, where individuals cluster into 3 columns along PC1. Indicative of 2 highly divergent haplotypes in their homozygous, like left and right, and heterozygous in the middle state. And when we combine this evidence with the LD stuff we saw earlier, we can infer that the divergence is maintained by chromosomal inversion, in this case, in Quillback. If I take a look at the PC1 loadings and isolate the 100 SNPs most responsible for separating individuals out along PC1, I get a pretty clear picture of who possesses which genotype. In this case, about half of Quillback are homozygous for what I'll call the Quillback haplotype. Many are heterozygous, and a few are homozygous for that haplotype that's more prevalent in copper. Next, I wanted to take some of the pure type individuals from each species, a handful of black and yellow rockfish samples, a close relative, generate a consensus tree. I annotated this a little bit, but the keen-eyed among you may be able to kind of already see what's going on here. All the copper individuals are clustered up here together. Fair enough. But so too are those Quillback that resemble copper, and with that very high sequence similarity, likely due to ongoing hybridization, balancing selection, and recombination, causing both species to look especially similar within that region. The other Quillback, possessing of the inverted haplotype, separate out all the way down there. maybe a little bit of incomplete lineage sorting here, with Quebec resembling their other close relative. Anyways, I did that in just about every one of those haploblocks on most of the chromosomes in both of these species, and found that the patterns differed depending on the haploblock, and the relationship between individuals was not constant in each of them. That'll all be summarized in my thesis, maybe sometime soon in an upcoming publication. With that, I'd like to thank you for your time. Hopefully you enjoyed the story. Hey.
Given that the LD is at the end of the chromosome, do you think it could be chromosomal fusion? Or have you measured LD between chromosomes?
I haven't measured LD between chromosomes, no, but that is a clever idea, and maybe one we can squeeze in before.
It's quite frequent in fish.
Pardon?
It's quite frequent.
It's quite frequent in fish.
And could even be polygamous.
Good to know. Thank you. Anybody else? Okay.
Do you have evidence for introgression versus incomplete lineage sorting or
Sorry?
do you have evidence for introgression or incomplete
Oh, I see.
sorting or
Yeah, we did some ABBA tests, and it seems like we have much more evidence for local introgression in those LD blocks than we do in genome-wide introgression. The signal is very weak for sort of genome-wide introgression between these, but I think that could be due largely to the fact that we use very species-specific individuals and not a lot of clear hybrids. Thanks.
So yeah, my name is Karolis Ramanauskas. I want to say a grad student, even though actually on the evolution website it says I'm a grad student. I'm a postdoc in Joe Walker's lab in Chicago at UIC. And this is the title of my talk, but I'm not going to talk about it. So yeah, so I got sidetracked and shit happened. So we'll see. Yeah. This is kind of an improv, so we'll see what happens. All right, but before I'm not going to talk about the thing, I'm going to talk about the thing a little bit, just to give you a motivation for it. So a couple of years ago, our lab, there's a primary author sitting there, Alexa Tishka. We published this paper where we identified, we basically downloaded all this is a strong word of chromosome, the chloroplast mitochondria for all the plants where both of them were available, and we only used the RefSeq. So that ended up being much smaller than what's actually, with the total number. And then we built phylogenies for each homologous loci and for each locus, and then we identified conflicts and significant conflicts that were supported by both trees in 4 parts of the tree. And then last year I did this thing where I just went for those 4 spots on the tree and tried to find exactly what's going on. And this is one of those examples. We have the same species on both sides, and then you have chloroplast tree with 62 loci and mitochondrial tree with 26 loci, and there's different topologies supported. And then we how many topologies support how many loci support which tree. So for chloroplast tree, most chloroplast supported. For mitochondrial tree, most mitochondrial loci supported, but then clearly not it's not 100% chloroplast support chloroplast tree, not 100% chloroplast loci support chloroplast tree, and same way for mitochondria. And then we look where on those genomes there are, how they're located, and there's some synteny and kind of a pattern of the thing, but there's some interesting things. Obviously, there's some of these our outgroups here is Arabidopsis, because that's kind of the only thing that we had that defined the node. But in general, even within within other species here, there's a lot of rearrangement happening in general. But so this was the thing. Now the thing is how did I, what did I do? And what I wanted to do again with this was to now go and get everything that we have. If we don't have a chloroplast or mitochondria assembled, let's assemble it. We have a lot of data every day. There's shit being uploaded to SRA. So we were thinking of just make it get a better grasp and do locus by locus, basically just do it very comprehensively. The thing is, before you can do that, and this is where things go off rails, is that this is kind of the expected workflow, and then, you have what actual workflow is. So basically, you download sequences, you extract CDSs, you get stuff, everything's great. The actual workflow is you get CDSs, and then you figure out that annotations are not always correct. Even if they are correct, people use different names sometimes. There's not strict validation when you upload stuff to NCBI, right? So sometimes you have a coding region annotated, but it's like, there's a, the first codon is not part of the, or, whatever. It starts at codon 2 or something. So you have to do like if you want to do it en masse, you do some sort of ORF detection, you like adjust for it. Of course, we're dealing with chloroplast mitochondria. Chloroplasts especially have a lot of RNA editing, especially when you get into like the ferns and stuff, you get a lot of RNA editing there. So the sequences that are uploaded actually don't have sometimes, reading frames that you would expect. You have to look, if the annotations exist, you have to look for, is there possibly RNA editing here? Is this the ORF, actual ORF somewhere there, right? So things like that. And then sometimes you have basically in the RefSeq even, you have genomes uploaded that are definitely from a different species than they're annotated as. So I have to obviously check for that. I had a couple of those examples. And sometimes you have things where you have like a chromosome. I didn't even know that at some point, that like mitochondria can have multiple chromosomes in mitochondria, but that's the case. So like in the RefSeq, for example, for one of the tomatoes, there's a chromosome 1, it's a RefSeq, great. And then like the rest of the genes that go on mitochondrial chromosome 2 or 3, they're all like from like, they're not in RefSeq, so not complete mitochondrial genome is there. So you kind of like, we were missing some loci in the past. I was like, how come there's like so few loci on some of these? And it's like, yeah, because there's other chromosomes that are not technically, they're not like, they're not annotated as such. So there's this process, and I got sidetracked with this, and there's some examples of things. I wrote some scripts to basically like use alternative names, kind of go through and like standardize the name. So like I can actually, when I end up annotating stuff, I can like just look for a specific gene or specific locus by name, and it's there, right? And sometimes people basically, there's some significantly different, like in a specific species, people will rename a gene for, I don't know specific reasons for that. I'm sure they have the reasons, and it's just that like for me, when you start looking for it, you start clustering them, like, so because the names don't match, like you cluster them by similarity to some degree, and then, okay, what's the most common name in this cluster? What's the most common name in that cluster? Stuff like that, and then you give it a name so you have some sort of database to work with. And some of them are just like that, like where it's literally just, annotator went and found open reading frames. Yeah, so in those in RefSeq, I just figured, that wouldn't be there, but no. So data is not great sometimes. I mean, it's there. I'm happy that it's there. It's just that we have to do some work. The problem is that you kind of have to do the same work often, multiple times, and you can forget. So like I was just kind of didn't know how to do this, because sometimes you have to literally go, like you have a bunch of sequences that are great, and then one of them, and you spend a bunch of manual work. It's a problem that you kind of didn't expect, or it's a weird misannotation that you like didn't write, that your script doesn't account for. So you end up working on one, and you really need it because there's only one for this particular species. You want to get that node, right? So ideally, also what we would want in general from these, like I want to have some sort of replicability, so I'm doing a bunch of these things. I'm pulling out pieces of the sequence, I'm editing them, right? Like to some degree, like I'm cutting pieces off. I want it to be documented. Like I want to know what I did, because like you do it to a lot of genes, you forget. Like you get, I want to know what versions of software I use, stuff like that. I want to know like those individual genes. If I use multiple accessions from the same species, I want to know like. Where the originals come from. Did I combine 2 accessions to make one from the same species, stuff like that, right? I also want to have the bibliography for things. Like again, when you're dealing with thousands of things, you kind of don't want to deal specifically, but then you may need to look up things, like if there's something wrong at this node, like let's go back to the source, see what's happening, does this look right? And for like slightly different workflows that for me, like phylogenetics and kind of, this kind of stuff, but I also kind of worked on some population genetic stuff. In general, like sometimes vouchers are really useful, just kind of knowing, because especially if the specific taxon you're working on, there's a lot of taxonomic changes happening. You may need to re-synonymize those names, so having some sort of taxonomic resolution that's like going through like specific people who only work on tomatoes or something, like they have much better database for that specific clade and so on. Like if you work on a specific like clade, you probably have a lot more information and you have much more understanding why things got renamed, and so mapping the tax IDs sometimes on GenBank, the name is old, but tax ID is correct, so you kind of have to do things like that. So basically, I just kind of I needed to make it simple for me, so I started working on a tool and basically stopped doing the actual work, but started doing the other work, So and my idea is to I want to simplify this process kind of for myself. And my goals were these are I wrote these goals today, kind of later. But that's I needed to put them on the paper now, and my principle is that I don't want to repeat myself. Kind of after I did, if I make a change, I want to have some sort of record of it that I can run a program and it will apply that record each time like a patch to the thing. So the idea is that I have a program that manages sequences, and edits to that. If I have a synonymy things, I have a database that okay, if you see this gene. And in this general case, rename it to this, just a standard name. Get the names from the database if you can. If there's a taxonomy resource that is better that used for specific clade, use that to get stuff. Acquire if you basically whatever if you need some sort of a tree a skeleton to build a bigger tree or something, base tree, you have, use Open Tree of Life for if you don't have a good tree for pieces, stuff like that. Use GBIF for location information and other data. And also keep track of them, so when you actually analyze a bunch of sequences, okay, I have these successions and these derived sequences from that, and I want to produce, this is my final alignment, also produce all the references that I can get from that, where these sequences came from, so kind of original authors get the credit for what they did, if or also just for myself, so I can go check something quickly if I need to find a PDF. So kind of slowly, so it slowly kind of bubbled into basically me writing this kind of GUI for myself, where I can search stuff. It's very minimal right now. So a lot of the work in the background is kind of on the database side. But I need to show something that I can that I've been kind of working on using for this. So so basically I have this kind of GUI for general you download a bunch of sequences, you get the information, you can organize them in different folders and stuff. I have basic sequence view, whatever. There is, kind of basic kind of management functionality. At this point, it's, just they have a bunch, different ways you can filter it by, this is an example of by different sequence type or source organelles and stuff. Filtering steps and so on. And this is really kind of a work in progress that I'm kind of building for to have an open source tool that I can implement, can do extensibility for it. So you would have a standard inputs outputs that you can write plugins for and deal with, and I can run pipelines. So my short-term goal, besides those really big goals, is to literally just have a thing that I can actually use quickly, instead of running Genius or paying for that license, just kind of be able to run math on a bunch of sequences, extract a bunch of CDSs, and just check, but the idea is basically that I have a system that I can, use to kind of produce a standard cleaned output. So again, at this point, it's barely functional, so don't expect anything. I do have a link and a QR code for my GitHub. You can check it. I compiled the version this morning, but I'm pretty sure it's not going to run on slightly older versions of Mac. In general, this will run on Linux and Windows too. I'm not at home right now. I will compile it for Linux too. I'm sure that will actually work. Macs are really capricious about this. And in general, I'm just looking for ideas from people, especially about other APIs and kind of things where you can get more information. So I would like to have some sort of central resource for let's say if you have interest in specific taxon of interest or specific locus of interest, you're working on a specific evolution of gene family or something. My idea is to have okay, for this gene family, give me everything that you can pull from the internet about it, obviously, as much as feasible. And yeah, I guess at this point, I'm kind of like figuring out where to focus specifically. I have plenty to do, just for myself, to make it usable for me, but in general, it'd be nice to have some feedback, Again, my contact information is from the GitHub. You can reach out to me if you have any ideas or if you have any needs. And, well, S disappeared. So that's my lab, simple acknowledgements lab. And this is Walker Lab and all the people in it. And I thank them, and I thank you for being here today. Thank you very much.
Okay. Well, hello, everyone. My name is Cisneros. I am a PhD student at Laval University, and I'm very happy to share with you this story we have been working on for a while now on how protein-protein interactions evolve after gene duplication. So first of all, I'm going to start with why study gene duplication. If you think of different model organisms, like, for example, human, yeast, and Arabidopsis thaliana, you can see that they have a high content of, their genomes are enriched in duplicated genes. So duplications continuously generate new genetic material that is able to evolve new functions that will contribute to the organism's evolution over time. So one of the motivations to study gene duplicates is to study their potential to generate new functions. But an important question in this sense is whether all gene duplicates have the same probability or have the same propensity to generate new functions. So, for example, here we study at the molecular level different kinds of duplicates. In particular, you can think if the ancestral gene codes for a monomeric protein, the two duplicates would be independent from one another. So they are able to accumulate different kinds of mutations. And in several scenarios, you can think that, for example, one of them preserves the ancestral function, while the other one is able to explore mutations that might compromise the ancestral function. But since the other copy is there, it can explore new functions and eventually specialize on those ones. But there are other kinds of proteins that form homodimers. So that is, they interact with another copy of themselves. If you duplicate those kinds of genes, the two duplicates will be able to interact with themselves, but because they are identical, they will also interact with one another. So this introduces a new interaction in the system, in the network, where you have the two proteins interacting with one another, and then whatever mutation occurs on any of the proteins will affect the respective homodimer, the self-interaction, and this interaction here. So we wanted to study what happens with this in this particular scenario. And in different references, there are many different outcomes. In several cases, the two duplicates become independent from each other because the heterodimer disappears. They go back to self-interaction, so they go back to being functionally independent from one another. In other cases, both the homodimers and the heterodimers are kept. And in yet another kind of cases, you only keep the heterodimers. So this becomes the only functional unit. And so, for example, in terms of adaptive hypothesis on how you get to these kinds of outcomes, you can imagine here, for example, they could specialize on new functions, or that here you could modulate the expression or the abundance of the proteins to regulate the formation of the dimers and then regulate function, that kind of thing. But this particular case is hard to let's say, rationalize through adaptive hypothesis. In particular, because now it introduces more complexity. Now you need two proteins to satisfy the role that the function that an ancestral protein was able to do on its own. So, for example, you can think, as I was saying, the ancestral protein used to form a homodimer that was able to provide some function, which could be, for example, binding to other proteins. But then if it is replaced by the heterodimer of the two duplicates, you need the two copies, because now if you delete one of them, you lose the functional unit, and then you lose the interactions to the other proteins. So the system as a whole becomes less robust. You need more parts now that are necessary for those functions. So the question we have here is, why does this happen often in evolution, and how do you get there? Is natural selection actually necessary to maintain the heterodimers and make them replace the ancestral homodimeric interaction? And to be able to approach these questions, we needed to know what is the neutral expectation. So what happens if there is no selection, no explicit advantage of one over the others, and how the system evolves as mutations accumulate. So we aim to approach these questions with a purely biophysical system. It's essentially a system where protein subunits are produced at a given rate. And then from the protein subunits that are available, and based on their binding affinities for each other and for the other protein, they would assemble into the three types of dimer. Importantly, right after the duplication, when all the parameters are the same, you would expect an equilibrium of 25% of one homodimer, 25% of the other, and 50% of the heterodimer. So it already takes an important part of the mix at equilibrium. And you can imagine that if you apply mutations that increase the binding affinity of, say, V for itself, this one will increase, and this one would decrease, and so on. So we want to study how this happens throughout evolution. And a very important thing is, as I was saying, we want to study the case in which this is neutral. So we want there to be no fitness, no direct advantage of one or the other. So say, for example, we are going to have a fitness function that will make fitness depend on the total activity of the system, and activity will be proportional to the number of dimers in the system, the concentration at equilibrium, regardless of the identity of the dimer. So all of them contribute the same. And so like this, even in this case where we have 10 copies of one dimer but nothing of the others, we have a given activity and given fitness, but this is the same as having 10 copies of, say, the heterodimer, or a balanced number of them. As long as total activity is the same, fitness is the same, so this is completely neutral. Activity will only change if the total concentration of dimers changes. So with the system, we wanted to set up simulations to study the evolution of the system. And to do that, we need protein structures, and we need mutational input. To generate this, we went to the protein data bank and surveyed structures of homodimers from the PDB. And then we used a computational tool called Foldex, which allows us to simulate effects of mutations on the protein structure. We simulated mutations, all possible mutations, single mutations on the structure of those proteins, both on one copy, which would be as the effect on the heterodimer, and on 2 copies of the dimer, which would be the effect on the homodimers. And this will allow us to have distributions of the effects of mutations on the energetic parameters of the heterodimer and the homodimer. So we have these distributions from which we can sample in the simulations. The way the simulations work is at each step, we sample one of the mutational effects from these distributions. We calculate the new concentrations at equilibrium, which will give us the total activity of the system and the fitness of the system. And from that, we get a probability of fixation from which we know if the mutation fixes or not. And then we continue, we keep going until 200 mutations have fixed. So now moving on to the results of the simulations. Here I show the results of simulations with 37 very high quality structures surveyed by another reference. And what we see here is that for the structures in the x-axis, most of them actually increase their concentration of heterodimers over time. So the y-axis refers to the percentage of heterodimers and homodimers at the end of the simulation, heterodimers in blue and homodimers in orange. And you can see that from the dashed line, the starting 50% heterodimers, most of them actually increase over time. So there seems to be something that actually enriches for the concentration of heterodimers for most structures, even though there is no explicit advantage. So we thought there has to be something in the distributions of mutational effects from which these simulations are sampling. So we compared the 2 most extreme cases, the one that goes the most toward heterodimers and the one that goes the most toward homodimers, to see what was different. So in fact, what we found at first is that they are very similar. So here I am showing the effects on the binding affinities of the 2 kinds of dimers. The x-axis is for the homodimers, so the self-interaction, and the y-axis is for the interaction between copies. Both axes, if the value is positive, it means it destabilizes the interaction, and if it is negative, it means it stabilizes the interaction. So as you can see, overall the effects are along the diagonal. Blue and orange represent the 2 most extreme structures. So blue goes toward heterodimers, and orange goes toward homodimers. And we see again that they occupy very similar spaces. But if you look very closely, you might see that there seems to be a bit of overrepresentation of orange dots above the diagonal and blue dots below the diagonal. Thinking about this, for example, a point around here would be neutral for the homodimer because it's at 0, but it stabilizes the heterodimer because it's negative. So this would be an example of a mutation that favors the heterodimer. And we thought, in this sense, because this is asymmetric, it is some sort of mutational bias that favors the heterodimer. So we calculated a function of the enrichment of available mutations that favor the heterodimer based on the magnitude of these deviations from the diagonal. And indeed, what we found is that this metric of enrichment of mutational biases that favor the heterodimer are highly correlated with the outcome of the simulations. When we have more mutations that favor the heterodimer toward the right, we see more heterodimers at the end of the simulation. So even if the magnitudes of these effects is actually very small, over the course of the simulation, these small effects seem to accumulate and drive the outcome that we see for the structures that we had. So we had a final question that we wanted to address, and it is, how do these mutational biases interact with natural selection? So we did a new series of simulations in which we would allow differences in the specific activity of the types of dimers. So the way this works is that if the homodimer is more active than the heterodimers, natural selection would naturally select for the homodimer and vice versa. And so what we see is that natural selection is very much able to revert the effect of these biases. So here we start at 0 with an enrichment of heterodimers. And toward the right, we have natural selection favoring the heterodimers even more. So we can see that they go to 100%. Toward the left, we favor the homodimers. So we see that they increase and cap at 50% each, so that together they make 100% of the dimers in the system. So with this, we concluded that natural selection is able to revert the effects of these mutational biases. So with this, I'll just do a brief summary of everything I've shown. So we show that natural selection is actually not required to preserve these heterodimers of paralogs, because they are expected to increase even in a neutral scenario where there is no specific advantage. And they could contribute to these cases where we saw that the heterodimer replaces the ancestral homodimer. We saw that this increase in the concentration of the heterodimer is associated with some mutational biases in the distribution of available effects. And we showed that natural selection can actually revert these effects if it is, strong enough. So with this, I would just like to thank my supervisor and my collaborators, my coworkers in the Landry Lab, the funding organizations, and you all for your attention. And I have the QR code to the manuscript if you would like to have a look. Are there any? Yeah.
Is there anything particular about the structure of the protein that more correlates favoring mutations?
Right. Yeah, that's a good question. Like, for example, like whether the mutations that are available depend on some structural parameters. Yeah, we tried having a look at this, but we didn't find any clear relationships. What we looked at was secondary structure content, symmetry at the interface, these kinds of parameters, but it wasn't clear. Like, we didn't find anything in particular. Well, thank you.
Hello, everyone. Thank you very much for coming to this talk. I'm going to present how amphibian MHC evolution may be selected by an emergent pathogen. So as we know that EID has been detrimental to our life and also economy, such as COVID-19. And in amphibian world, there's also an EID. It's called chytridiomycosis, and it's caused by chytrid fungus. So one of the chytrid fungus is BD. So here are the BD-infected frogs found to die in the wild, and here are the BD-infected frog in the lab. So where did BD come from? So, a few years ago, the science paper suggested that BD originated from Asia. What is interesting is that in Asia, actually the Asian amphibian somehow appeared tolerant or resistant to BD infection. So then I tried to understand why. with the Korean amphibian species when I was a PhD student. So what I have found is that the Korean amphibian species actually that carry multiple hypervirulent BD lineages, but they have evolved resistant or tolerance to BD infection. Then I try to understand the mechanism behind the resistance or tolerance. So previous research has suggested that BD resistance is related to MHC class 2 genotypes. So what is MHC2? MHC2 can recognize the peptide derived from the exogenous pathogens and present it to T cells. And then the adaptive immunity is activated. So usually MHC class 2 is selected for pathogens, and it's extremely polymorphic, and the polymorphism usually reflected in this beta 1 domain. So in this research, I use these two species, this 100% BD resistant species from Korea, and this about 90% susceptible species from Australasia. The MHC, then I genotyped MHC and compare the genotypes. The MHC genotyping I used here is RNA-based instead of genomic DNA-based, because RNA-based MHC genotyping can cover the full length of beta 1. The first interesting finding is that the glycine 90 beta 1 deletion, which is found in a much higher proportion in the BD resistant species compared to the susceptible species, as you can see here. And every individual from the BD resistant species has MHC alleles with this deletion. And the statistical analysis supports that BD resistant is positively related with this deletion. In one of the two BD resistant individuals from this species, from this susceptible species, has this only allele, and this only allele is only found in this resistant species, in this resistant individual, and this allele also have this deletion, as you can see here. This suggests that this allele may be a rare allele selected for BD resistance, and the similarity shared between the two species is called trans-species polymorphism. Trans-species polymorphism. This TSP usually has two resources. One is two resources. One is from the same ancestor. Another reason it could be convergent evolution. For this, in this case, I found it's followed convergent evolution because although they share the same amino acid deletion, the two species use different codons leading to this deletion. The details can be found in this paper. And another interesting finding is that the MHC alleles with this deletion are under significant positive selection in the BD susceptible species. Other selection tests suggest that more positively selected sites in the susceptible species compared to the resistant species. And among the positively selected sites, as you can see that here, this is site 9, 12, and they are this is possibly selected sites in the BD susceptible species completely conserved in the BD resistant species. And then I try to understand the physiochemical property. Physiochemical property of this sites suggests that hydrophobicity is most likely selected for BD resistance. Next, I try to understand how the genetic difference between the two species. What I found is that in the BD-resistant species, a potentially peptide-binding sites, it's called WUCA sites. It has a much higher genetic distance, suggesting a divergent allele advantage in this BD-resistant species. Because MHC diversity usually related with disease susceptibility and selected by various pathogens, I try to understand whether MHC diversity is associated with BD resistance. What I found is MHC alpha diversity is not related with BD resistance, and because the number of MHC alleles and supertypes are similar in the two species. However, beta diversity of MHC supertypes is significantly different in the two species, suggesting that specific functional MHC supertypes, like this one, might be selected for BD resistance. So to sum it up, what I found in this study is that there's a potential rare allele with the G90 beta-1 deletion in the BD-resistant individual from BD-susceptible species, which might reflect the early stage of MHC evolution to an emergent pathogen. And the more divergent MHC2 alleles found in the Asian BD-resistant species maybe reflect the later stage of MHC evolution to an emergent pathogen, to BD infection. To understand it simply, as you can see from this model, when in the early time of this MHC evolution upon emergent pathogens, rare allele could be selected, and when the pathogen driving force is high, and when the rare allele is fixed, and when the pathogen driving force is low, the divergent allele advantage is most likely to be detected in the resistant species. And when new emergent pathogen appeared, and it's most likely the rare allele is selected again, some new rare allele, and when the rare allele is fixed, and when the driving force by pathogen is low, it's likely to be detected the divergent alleles in resistant individuals again. So this scenario may continue. And also in this study, I found hydrophobicity in certain amino acid sites is most likely to be selected to defend BD infection. And the MHC alpha diversity is not selected for the resistance in Asian BD resistant species, but it's most likely specifically functional supertypes is selected for BD resistance. Yeah, maybe I talked too quickly. And thank you for your attention. And I would like to thank my collaborators and advisors and all the co-authors in the papers and all other researchers in this field who contribute to all the MHC studies to understand the role of MHC. And also my funding from Korea. I would like to give a special thanks to John, who guided me through the MHC genotyping, which is somehow complicated but extremely important. Thank you. I would like to take your questions.
You said that the species that is susceptible is only 90% susceptible.
Yeah, yeah.
So what do you think that 10% is coming? Is associated to a different molecule, or do you think that maybe some of the uptypes that you are seeing in this MHC class 2 are helping to that resistance?
That 10% maybe like accidentally they adapted to the BD infection. So, did I get your question? Can you repeat it?
Yes.
It's kind of a similar question. I may have just entirely missed this. You talked about species being resistant or individuals being resistant or susceptible.
Yes.
Did you do controlled infections to document that, or is it just that they seem to do okay in the natural environment?
Oh, sorry, I did not talk very clear. Maybe I talked too fast. I did not explain. So, for this species, for this littoral white frog, it was naturally found highly susceptible in the field. But in this species, it's not 100% susceptible. Some individuals really, like in my experimental, like laboratory study, I found only about 7% Littoria survived from the BD infection. So that means 93% completely susceptible from it. And yeah. And for the Korean species, usually we don't find a mortality or morbidity in the field. And so I infected, so to prove that they are really resistant or tolerant, I did infection experiments in the lab, and I treated a really high dose of the chytrid, and then what I found is they all survived from the chytrid. Some died, but it's not because of BD, because the BD load is really low.
I guess it's just the fact that you have one individual with a rare genotype that seems to do pretty well. Obviously, experimentally, it would be kind of interesting to see if you could produce offspring. Is it something that's repeatable? So I don't know anything about your experimental system, but obviously that would be a direction that you could go if that was tractable, which would be a lot more compelling, right? That you'd be able to show that, yes, I can predictably produce genotypes that are resistant.
Actually, that would be really important if this study can be continued like with more generations. But the problem is, the problem in this kind of study is that after wild caught, somehow it's difficult to get more generations. It's just one generation, so we don't reproduce it. So it's just transient. So it's really, I think really more species should be tested whether like a similar pattern can be found for BD resistance. And it's not only one individual survived, it's 2 individuals survived. Yeah, I try to present as much result as possible, so I did not tell the details. So among the 30 individuals, Littoria I treated in the group, 2 survived. So among the 2 survived, 1 was sacrificed for my senior's experiment, so I could genotype the MHC using that, from the label, and the other survived individual. I did not kill it, so I could not get the liver. So I swapped the buccal and genotyped it. So that individual, it only has this one MHC, this only the allele I mentioned, and it could be tissue specific, but because it's the only one allele I got from that individual, and it also has the deletion, so somehow, it could be rare allele, but it's very, it could be wrong, but yeah, I think it's possible. That's it. I couldn't prove it. Yeah. Since I talked very quickly, if you have more questions,